Okay, good afternoon, everybody, and welcome to the monthly seminar for the Gulf Coast Consortium on Antimicrobial Resistance and the Center for Antimicrobial Resistance and Microbial Genomics. Today, I have the pleasure to introduce Dr. Jeff Tabor, who is professor at Rice University, and he's going to talk to us about synthetic biology approaches to studying bacterial two-component system. Jeff, all yours.
Great. So thanks, Cesar, for the invitation, and to Adriana Rosado, who also invited me. Yeah, so I'm a synthetic biologist. So I genetically engineer cells for useful purposes. And so I'm going to talk today about some technologies that we have developed that are enabling us to better study bacterial two-component systems, which are an important family of signal transduction pathways in antimicrobial resistance. So here we go. So an introduction to bacterial two-component systems, if you're not already familiar. Typically, these systems are comprised of two proteins. One is a transmembrane sensor histidine kinase. This protein is typically produced in an inactive state, and it typically binds to a chemical stimulus or even senses a physical change. And when it does, the sensor domain that's outside the cell becomes activated, undergoes a conformational change, and that conformational change is transmitted across the membrane into a cytoplasmic domain called the histidine kinase domain. This domain phosphorylates itself with ATP, and then it transfers that phosphoryl group to a second protein, which is diffusing in the cytoplasm, called a response regulator. Phosphorylation of that response regulator typically causes it to dimerize at a so-called receiver domain, REC domain. That's the phosphorylation domain. So it's like a switch where it goes from monomer to dimer upon phosphorylation. And that brings together two DNA binding domains, which are appended to the C-terminus of the response regulator, allowing them to sit down on a piece of DNA, which is typically a direct or inverted repeat operator site. And then the response regulator typically recruits RNA polymerase and activates gene expression, though it can also repress gene expression. So this is a simple signal transduction pathway, and bacteria use this architecture to sort of link changes in the extracellular environment to changes in gene expression. And so what's really interesting about this family of pathways is that they are highly evolutionarily diversified, and they are highly evolutionarily spread through bacteria. So to the diversity, these sensor domains can sense sort of a remarkable range of inputs, from metal ions of particular oxidation states to amino acids or peptides or proteins, to monosaccharides or oligosaccharides or polysaccharides, quorum sensing autoinducers, hormones from humans or plants like adrenaline and auxin. They can sense physical things like membrane stress, changes in temperature, or even light, and a wide range of other things as well. In terms of what they control really important bacterial behaviors like virulence, gene expression, antibiotic resistance, gene expression, biofilm formation, quorum sensing behaviors, things like bacterial metabolism and respiration, survival inside of macrophages, factors that allow gut colonization, and stress response pathways, among others. So this is the largest family of multi-step signal transduction pathways in biology. We have identified over 25,000 two-component systems, non-redundant two-component systems in bacterial genomes. And about 99% of these pathways are uncharacterized, right? So we know a lot about them from a couple hundred that have been characterized, but it's literally the tip of the iceberg. There is a huge mountain of these pathways that we know literally nothing about other than that they're two-component systems. So my lab has done some work in the past to begin to engineer these systems for different applications. So initially, we began with an interest in optogenetics, and we did a lot of work to engineer bacteria to see light using two-component systems. So we identified two-component systems from photosynthetic organisms like cyanobacteria or purple photosynthetic bacteria. And we have developed synthetic biology methods to functionally express these pathways in E. coli and in Bacillus subtilis. So E. coli being the leading gram-negative model organism and Bacillus subtilis being the leading gram-positive. We've expressed 4 different light-sensing two-component systems, sensing wavelengths from the ultraviolet all the way to the infrared, so across the UV visible near-infrared spectrum. In doing so, we've learned a lot about how to express these things heterologously, how to make them respond with large responses, and even how to re-engineer them, which I'll talk about shortly. We've had some applications of that bacterial optogenetics work. So we've done spatial control. So this is what we call bacterial photography, where we shine a pattern of light and dark across a petri dish of engineered bacteria that can sense light quantitatively, and get them to control a pigment producing enzyme with the light sensor, and then they physically print out the image that we shine on them. So this is a way to do quantitative control of bacterial gene expression across a population. We've engineered more sophisticated bacterial population behaviors like multicellular pattern formation, where they perform logic on the light dark signals that you're sending them, and then communicate with one another to actually form patterns that are not the same as the light signal that you shine on them, but are a result of it. So this is edge detection, as we call it. We've done a lot of temporal control, where we study the input-output kinetics of two-component systems. So what happens when you increase the light intensity or decrease the light intensity? How does that change the two-component system output in time? And then we've used that information to actually program gene expression signals such as sinusoids or step functions in response to time-varying light signals. Finally, we've done some recent work with Meng Wang over at Baylor College of Medicine to actually use light to control gut bacterial metabolism inside of the gut of C. elegans worms, for example. So here we're using light to stimulate E. coli in the gut of C. elegans to secrete colonic acid, which is an exopolysaccharide that Meng has discovered it improves mitochondrial health and increases longevity, among other benefits to the worm. So those light sensors have interesting applications. We've also done some work to engineer diagnostic gut bacteria. So here we discovered a new two-component system in the genome of a marine bacterium, Shewanella halifaxensis, that senses the oxidized sulfur molecule thiosulfate. And we were interested in that molecule because we thought it might be a biomarker of colon inflammation. And it turns out it is, at least in the animal model we're using. And so we discovered this new two-component system. We expressed it in a probiotic strain of E. coli and got it to turn on green fluorescent protein. We then administered those engineered E. coli to healthy mice and to mice who had colon inflammation. We collected the fecal samples 6 hours later and ran them through a flow cytometer, and we could reliably diagnose the extent of inflammation that the mouse had in its intestine with our bacteria because they became more green due to activation of this two-component system. So these are some examples to show you some of the reasons why we've been interested in two-component systems from a synthetic biology standpoint, from an engineering standpoint. They're very nice biosensors for useful inputs and can be used for a lot of reasons, in addition to being involved in antimicrobial resistance and virulence. And what we found in some of our work is that there are still a lot of challenges to harnessing two-component systems. So I told you less than 1% of these things are characterized, right? And so if you go into a typical genome from a typical bacterium, you'll find about 25 two-component systems. And you can identify them from sequence because the sensor histidine kinase gene typically sits next to a response regulator gene that phosphorylates. They typically are within a couple hundred base pairs of one another. And so you can identify about 25 pairs of those in a typical genome. But in almost every case, you have no idea what the sensor domain senses based on its sequence. And you also have no idea what promoter site the DNA binding domain of the response regulator binds to, right? So you can see the system, but you don't know its input and you don't know its output. And that system is pretty hard to work with given those limitations. Another limitation is that, as you probably know, you can't culture most bacteria in the lab, right? And so you can't do classical genetics on these two-component systems, most of these two-component systems, because you can't grow the bacteria or you can't make the knockouts and then can't do the screens. And so that makes them hard to study. I showed you some examples where we ported two-component systems between bacteria, and that works sometimes, but sometimes it doesn't work. And it appears that there's incompatibilities between the response regulator or the output promoter of a two-component system and the RNA polymerase machinery, transcriptional machinery of a non-native host bacteria. And these can take on various forms, these incompatibilities, and they take a long time to figure out what's going on in each case. So each case can be a unique case. It can take, a Ph.D. to figure out what's the problem. And so it makes these hard to work with. Right. And then finally, there's a lot of regulatory crosstalk. So these are environmental sensors, right? And it's, as engineers, we like to think one input, one output, linear sort of sensing pathway. But in reality, bacteria are actually integrating a lot of environmental signals through these two-component system output promoters. So you'll often have multiple secondary, tertiary regulatory signals that are activating or repressing your output promoter from your two-component system. And conversely, you'll often have a situation where your two-component system will activate, say, a couple hundred genes in the genome, not just the gene that, the primary output promoter that you're studying, but many others. And you may not know that this crosstalk is even present, right? And untangling this crosstalk can take one or multiple PhDs for a two-component system, right? So again, you're in this scenario where you've got sort of a lot of confounding factors to sort of just doing simple assays with these things and figuring out, their primary function. And so there have been a couple efforts to try to, that could be used to overcome these problems in the literature, and I'll talk about them briefly. So the sort of classic two-component system engineering effort started about 30 years ago with a paper from the Inouye group in Science, where they swapped the sensor domain of one two-component system, one sensor kinase, onto the histidine kinase domain of a second two-component system, right? So in this way, say you wanted to study a sensor domain, but you had problems with the output promoter of that system. Well, you could just sort of rewire your sensor domain onto a really well-characterized two-component system that didn't have a lot of crosstalk, had a really, well-behaved output promoter and so on. That would give you a nice assay for how that system works. And this works sometimes, but one of the major problems here is that sensor domains are really diverse and their mechanisms of activation are really subtle in many cases. Subtle conformational changes that are not conserved across a lot of sensors. And so we don't have any general rules for how to do this. If you just take a sensor domain and try to chimerize it with a different pathway, you're unlikely to be successful, generally speaking. And this has been borne out in the literature. There have been only a handful of successes in doing this in 30 years, despite quite a bit of interest, including from ourselves in taking this approach. Right? There's another approach that came out of Mike Laub's lab at MIT about 10 years ago. Jose Onachik has done some work on this computationally at Rice, where you could instead just rewire the interface between a sensor kinase and a response regulator. So here, you wouldn't have to worry about making a chimeric kinase, or at least a chimera between the sensor and kinase domains. Instead, they would just slice off a smaller piece of the histidine kinase protein. There are about a couple alpha helices here that are involved in an interaction, protein-protein interaction with the response regulator. So you could just cut those off and replace them with the alpha helices from a better or a well-characterized response regulator output, and sort of rewire your two-component system to a good promoter that way. So this works. It works especially well at just driving phosphorylation from a kinase to a response regulator, right? But what it has not been able to do yet is actually recapitulate the full function of a two-component system. So in other words, the chimeras that have been built using this interface rewiring approach are not responsive to inputs. Not responsive to the native stimuli. So this approach is sort of not yet ready to be used to study the pathways, generally speaking. The final approach came from John Duber's lab in Adam Arkin at Berkeley. And this is a clever approach where you just take your histidine kinase and you want it to talk to an artificial response regulator. Well, why not just put a protein-protein interaction domain, a bait on your kinase and a prey on your response regulator, for example, and just make the proteins, put them in close physical proximity, and they will tend to talk to each other, even if they didn't evolve to talk to each other specifically, because there is some general conservation in the shapes and active sites of kinases and response regulators. And this approach works. It does require a lot of tuning of individual systems because the interactions can be too strong and too weak. They can have lots of unintended effects. And this approach still suffers from the same problem of the Laub approach, which is no ligand responsive or input responsive two-component system has been engineered. So this can drive phosphorylation really well, but it can't recapitulate the full function of the pathway. So it doesn't really help you out. So a few years ago, a postdoc in my lab, Sebastian Schmidl, who's now a professor at Texas A&M, had an idea that we could look at a different location on two-component systems to engineer them. So in fact, or in particular, the DNA binding domain. So could we leave the full sensor kinase intact, leave the phosphorylation domain, the REC domain of the response regulator intact, and then just cut off the DNA binding domain and put a synthetic DNA binding domain from a different two-component system there, and its cognate output promoter, and make a chimeric pathway that way. And we thought that this approach might have some advantages. In particular, if you look across nature, these DNA binding domains, which are the major type of what's called an effector domain. There are other types of domains that can go in that position. DNA binding domains are the major one, but they're quite modular. So you look and you have a couple large classes of DNA binding domains. Up here, the AMPR-PHOB, the largest class of effector domains. Then you have the NAR-L-FixJ. These are the second largest class of effector domains. But then you just get a lot of different folds that evolution has placed in that position on that protein, right? And they can all be activated by phosphorylation of the response regulator receiver domain. And we think the reason why, is that there's kind of a core activation mechanism of response regulators that's very simple. Right, and it doesn't require a lot of evolutionary fine tuning in order to work. Right, so this is just some structural examples of response regulators from the AMPR-PHOB subfamily of response regulators. This is the largest subfamily of response regulators, and when they're not phosphorylated or when they're in the inactive conformation. These response regulators are monomeric, and they take on, different structures. So this is the REC domain up here in blue and yellow. And this is the DNA binding domain down here in green, in red. And you can see there's, they vary from no interaction whatsoever between the REC and the DNA binding domain to extensive interactions between the REC and DNA binding domain. But what's interesting is when they get phosphorylated, when the REC domain gets phosphorylated, they adopt a common activated structure where the REC domain forms a dimer. And the DNA binding domain is then connected by, by a flexible tether. So there's a flexible linker between the two domains, and then it's sort of free to go sit on its direct repeat or inverted repeat operator site. And so, despite the diversity of the inactive structures, these things are able to sort of undergo this common dimerization. And, just the act of dimerizing two DNA binding domains increases their affinity for DNA as the square rather than, so exponentially rather than multiplicatively. And so it's a strong increase in binding affinity in response to dimerization. And it appears that this alone, just dimerizing these things, can cause them to bind DNA much more strongly. And so that simple mechanism sort of may be able to be conserved if you just go in and start putting artificial DNA binding domains in here, right? As long as they're monomers when non-phosphorylated and dimers when phosphorylated, maybe that's good enough for activation. So we wanted to kind of test this hypothesis. So this was done by Sebastian and a grad student in my lab, Felix Eknes. And what they did is they took our red light sensor pathway, I mentioned it before, and this pathway, the histidine kinase, is turned off in response to a red photon, and it's turned back on in response to a far red photon. And the response regulator in this two-component system is AMPAR. It's the namesake of the AMPAR Phoebe family. And we sort of made chimeras between AMPAR and the response regulator from CCAR. CCAR is comes from a totally different two-component system, from a totally different bacteria, but its structure is of the AMPAR family. So these two response regulators are structurally related, but functionally unrelated. Okay, and so we made these chimeric pathways and we linked them all to green fluorescent protein. So we made actually 39 different crossover points. So everywhere you see an arrow, so here's an alignment between AMPAR and CCAR. This is the REC domain, the linker, the DNA binding domain. And everywhere you see a little triangle, that's where we made a chimera. So we stopped the AMPAR sequence and carried forward with the CCAR sequence at that point. So we made chimeras kind of all throughout the second half of the REC domain, and then we focused a lot of chimeras near the linker because we thought that would be a hotspot for getting these chimeras to work. And then we tested all of their responses to red light. Right? So there's a lot of data here. If you just look at the bars on the top, this is the fold change, the fold activation and the activating condition versus the deactivating condition for each chimera, each of the 39 chimeras. Right? So off on the right, here's wild type. The wild type pathway in this context is activated about threefold. And what you can see is there's actually a lot of functional chimeras here, activated, reproducibly activated by 50% or greater. And what you can see is that the activity gets better the closer you get to the linker, generally speaking. There's an optimum crossover point here just after the linker region, right at the beginning of the DNA binding domain. So that was pretty encouraging, especially the fact that multiple chimeras work. So if you've ever tried to make a chimeric protein with any kind of, sort of where the function of the protein was the sum of the parts of the two domains, you know that it's you usually don't get multiple successful chimeras when you try to chimerize something. There are often very subtle structural rules for how to make a functional chimera that you must obey in order to get it to work. And there's not kind of a general cut and paste capability, a plug and play type capability where multiple crossover points will give you functional proteins. And so the fact that we got 10 functional crossover points here on the first try was pretty encouraging. So we focused in on that linker region. we picked one amino acid just right before the linker, one amino acid right after the linker, and we rewired this pathway to 4 additional DNA binding domains. And these all come from the AMPR-FOB family, so PSDR, YXDJ, BCER, MANR. In fact, several of them come from antimicrobial resistance pathways. You may recognize their names. And we just picked them because they're well characterized. They had well characterized output promoters. And we got those promoters to work in E. coli, our model organism, even though none of these pathways came from E. coli. And so what we found is that about 4 of the 5 DNA binding domains that we tested work. They can be chimerized with AMP-R, right? And in particular, we didn't have to build a library for these additional 4. We just picked some of the best crossover points near the linker, right? So this means you could have sort of a simple approach to do this without building a library. We then wanted to see if the results generalized across the family. So we went to another AMP-R-FOB response regulator, which in this case, TOR-R. This comes from a trimethylamine and oxide sensing 2-component system from E. coli, TOR-SR. And here's its wild type response to the wild type pathway to TMAO, the input. And then we just made chimeras with all 5 of those same DNA binding domains that we had tested with AMP-R. And what we found is that 3 out of 5 of them worked. And in particular, this position right after the linker, position 137, was a really good amino acid to cross over TOR-R to synthetic DNA binding domains too. And again, one of our synthetic systems, PSDR, actually worked better, had a larger fold change than the wild type pathway. Right. So what this data showed is that, this result is general. We didn't just sort of get lucky with two component systems that could be chimerized with each other, but it looks like, multiple DNA binding domains can be rewired to multiple 2-component systems from the family at standard amino acid crossover positions. So this will allow us to sort of generally exploit this method to study these systems in the future. So very briefly, we repeated this with the 2nd largest response regulator subfamily. So this is NARL-FixJ. These response regulators have slightly different structures than the AMP-R-FOB. In particular, they've got another alpha helix in the linker. So it's not just a totally floppy linker like AMP-R-FOB, but there's an alpha helix, and it actually can interact with the other domains in the protein. So we thought we may have more trouble because there's more structure here in this family, generally speaking, but we did not. So we took E. coli, NAR-XL, which is NAR-L, here's the response regulator, and it's the namesake of that family. And we rewired NAR-L near its linker to YDFI, which comes from Bacillus subtilis. It's involved in an antimicrobial resistance pathway as well. But again, we just took it because it had a well-characterized promoter. And we were able to get about 9 different crossover points that we tested out of 11 or 12 that we tested were functional. They gave functional chimeric pathways. And so this looked like this family was pretty amenable to rewiring as well. We went on to show that we could do it with other DNA binding domains, not just YDFI, but LIA-R, UHPA. So these are other well-studied DNA binding domains from this family. And we could even do it with a second 2-component system that has a response regulator with the NAR-L subfamily. Right? In particular, site 159 right here, right at the beginning of the DNA binding domain at the linker or after the linker. This worked in every case, in every chimera that we tested with multiple DNA binding domains and multiple 2-component systems. So what we have now is for the 2 largest families of subfamilies of 2-component systems, AMP-R, FOB, and NAR-L-FixJ, which comprise about 75% of 2-component systems that activate transcription. We have what we think are standard output modules. So standard DNA binding domains, standard amino acid, and a standard output promoter that you can just plug in to a 2-component system from that family and have a reasonable chance of being able to wire it up to your, to a well-characterized output promoter so you can study it. And so it's a pretty general method for studying these pathways. We wanted to show why it's general. So I've sort of told you the limitations, but we wanted to demonstrate that. And so here's one benefit, is that this rewiring is just a really simple way to eliminate cross-regulation that can be confounding your experiments. So TOR-SR, this two-component system, it actually has an accessory protein here, TOR-T, and the kinase actually has some extra domains, extra phosphorylase steps. That's not important. What's important is it's activated by TMAO, and it activates its output promoter, but that output promoter is dominantly repressed by oxygen, right? And so E. coli do this because if oxygen is present, they don't want to make the enzymes to respire TMAO. They don't want to make ATP using TMAO. They want to make it using oxygen because they get more ATP from oxygen. And so that's biologically why that's there. And so you can see that experimentally. If we grow bacteria with the wild type TOR-R and the wild type output promoter in the absence of oxygen, we can stimulate this pathway, high levels of output activity. But in the presence of oxygen, we see about a 10-fold repression of the pathway by oxygen. And then we just did that same simple test with each of our chimeric pathways. So these, I'd showed you a couple slides earlier where we rewired the TOR-R to CCAR and its output promoter, PSDR and its output promoter, YXDJ and its output promoter. And as expected, we abolished this oxygen repression, right? And so this is kind of an obvious result. I mean, these output promoters are not known to be repressed by oxygen, so this is what you would expect. But in most cases, for most two-component systems, you don't know that the cross-repression is there, and you can't predict it, right? And this cross-repression is known because this pathway has been studied for decades, right? And a lot of PhDs went into studying that oxygen repression, right? But we don't have to have that. We can just get rid of it, even though we don't know it's there. And we don't have to learn about it in order to study the pathway itself. So that's, we think, a benefit here. Another major benefit is moving two-component systems into heterologous hosts where they can be studied more easily and in isolation from the networks that are cross-regulating them. And so actually, if you look in the literature, there are very few examples of someone moving a two-component system from a gram-negative to a gram-positive host, right? And one reason that this, you might think this could be, is that the membrane lipid biochemistry is quite different between gram negatives and gram positives, and maybe these sensor histidine kinases don't, they've evolved to work in a gram negative membrane, but they just don't work in the gram positive membrane, maybe something like that, right? So we took this nitrate sensing two-component system that had evolved in E. coli, and we expressed it in Bacillus subtilis, gram positive, and we connected it to GFP and looked at the output of the pathway in the absence of nitrate and in the presence of nitrate. And what you see is it has very low activity. So this 100 methyl units, methyl is molecules of equivalent fluorescein. So it's a calibrated fluorescent unit. So this is very, very low methyl activity. It's basically bacterial autofluorescence. Okay, so they're not getting activated by nitrate. So we just swapped the DNA binding domain and output promoter for one of our standard output modules. We put YFDJs, or YDFI, sorry, DNA binding domain onto NARL, and we gave it its output promoter, YFDJ, and those had evolved in Bacillus subtilis, right? That's actually where they originally came from. So we knew they would work in Bacillus subtilis. And so by doing that, you can see the data on the right. That's the only modification we made. It was the first one we tried, and we got a 2,000-fold activation in response to nitrate. Right, so the two-component system from E. coli works robustly in Bacillus subtilis if you give it an output promoter and DNA binding domain that work in Bacillus subtilis. So we actually think a lot of the problems when you try to move these pathways around is this incompatibility or poorly understood regulation of these output promoters, and this is a nice way to get rid of it. Okay, checking the time. All right, so finally, this is why we think the method is most important, is that you can actually discover two-component system inputs, right? So understanding why bacteria use these pathways biologically, what they're sensing and responding to, is critical, and it's very poorly understood. So we wanted to show that the method can enable that discovery. And so we picked a model organism, Shewanella onadensis. This is a freshwater bacterium, and it's famous for being able to breathe all sorts of metals in the absence of oxygen, right? Uranium, all sorts of crazy metals. And it's known that two-component systems sense metals outside of the cell, or they sense electron acceptors, which metals act as, which allow bacteria to change their respiratory metabolism, right? And so we sort of thought we were giving ourselves a leg up. We'd try some of the metals as inputs against the against novel two-component systems from this organism. So that's why we picked that organism. And we identified 8 two-component systems in its genome of the OMP-R-Pho-B subfamily that were homologous to nothing. So these, we could not predict what these sensed based on their sensor domain or any other aspects of their sequence. And so they were totally uncharacterized. So we expressed each of the, we actually got 7 of these things to clone into E. coli. We expressed each of the sensor kinases alongside the rec domain of their corresponding response regulators, but we rewired all of them to our PSDR DNA binding domain and its output promoter. And then we linked them all to GFP, right? So all pathways were chimeric here. And then we did a big screen, right? So what we did is, if you look on the horizontal axes here, you'll see a bunch of chemical names. And so we assembled a list of the metals I mentioned before, but also various other amino acids and chemicals that, you know, bacteria care about, generally speaking, or we thought they would care about, and that were sort of readily available in the laboratory, right? And then we tested the response of each of these chimeric pathways to each of those 120 chemicals, right? And so on the vertical axes, you're looking at different two-component systems. So the one on the left is TOR-SR. That's a control. We know that senses TMAO. We did the screen, and you'll see there's one, you know, bright blue spot. That's a 10-fold activation in response to TMAO, and it responds to, you know, nothing else, or nothing else so strongly, right? And so that told us that our screen worked. And then we did that for all these uncharacterized two-component systems, and we got some hits. You can see some intense bands in there. And we actually did, after the initial screen, we did quite a number of controls to actually examine what was happening with a lot of these things that were giving weak activation and weak repression. And we were able to quickly narrow it down to sodium hydroxide. So if you look down at the bottom, the third uncharacterized two-component system, 4387, 4388, that thing is turned off by sodium hydroxide, by basic conditions. And it's actually turned off by a bunch of other things that we had to dissolve in sodium hydroxide. Okay, and so we hypothesized this was a pH sensor. And so we went on to test it. And so what you're seeing is here on the left is a little domain map of this novel sensor histidine kinase. It has 2 transmembrane domains and a protruding extracellular sensor domain, sort of a classical looking two-component system. And then it has the 2 domains inside, the DHP and the CA domain. These are part of the histidine kinase domain. And so we did a sort of a pH curve. So we went from basic to acidic. And what you can see is we get a nice activation, continuous activation of this two-component system as we cross neutral pH, basically. So it basically turns on at acidic pH and off at basic pH. And then when we mutate any of the phosphosignaling residues in this two-component system, so the histidine, the catalytic histidine in the histidine kinase, we abolish the response. And if we mutate the phosphoaccepting aspartate in a response regulator, we also abolish the response. And so we are pretty confident that this is a novel pH sensing two-component system that had not been reported before. And one thing that's interesting is this is conserved in some other gram-negative pathogens. Actually, it's conserved in Yersinia pestis, the causative agent of bubonic plague, where it's also uncharacterized. It sits next to what appears to be a secreted extracellular aggregation protein. I think the bigger point here is not that we found a pH sensor, kind of surreptitiously here, or serendipitously, sorry, but we actually developed a method that makes it easier to make these discoveries, right? To actually discover, we can just do high throughput screens on systems to more readily discover what it is they're sensing. So where we're taking this now is we're scaling up and studying the two-component systems of the human microbiome, right? So we've done some computational work. So we analyzed about 460 high-quality human gut bacterial genome sequences from the Human Microbiome Project. We identified all the two-component systems in there that we could identify, and we put some criteria to sort of increase diversity and decrease redundancy. And we ended up with a list of, and then we enriched for, or we selected all the OMP-R and the NAR-L two-component systems among all gut bacterial two-component systems. So we ended up with a list of 543 sort of prioritized two-component systems. They're all uncharacterized, basically. We don't know what any of these things sense. We just know they're from gut bacteria. Most of them are OMP-R family, two-component systems, just how the numbers worked out. Some of them are NAR-L. And so what we did is we actually synthesized the DNA for all 543 of these pathways and rewired them all to synthetic DNA binding domains and all to such that they all expressed a barcoded messenger RNA that could be read by next generation DNA sequencing. And they also controlled GFP, so we could measure the activity. So just briefly, the way this works is we had a standard E. coli vector backbone here, and we expressed each of the sensor histidine kinases from that backbone. And then we co-transform that plasmid with the sensor kinase with its particular response regulator. And here we introduce that response regulator's rec domain under a promoter. And then we use our standard crossover point, so position 137, to remove the DNA binding domain that evolved onto each response regulator and replace it with that of CCAR, one of our standard output modules. And then that activates the CCAR output promoter in GFP, as well as a barcoded RNA. Okay, so that's how it worked. It was a lot of DNA synthesis. And so we did this in collaboration with Caribou Biosciences, a company in the Bay Area. And worked with Rob Britton at Baylor to actually develop an assay for measuring the response of all of these two-component systems to human fecal samples, right? And so we basically collect the samples, reconstitute them in media, add the fecal sample to a culture of bacteria expressing all 500 plus two-component systems. We incubate for an hour, allow the two-component systems to respond to the fecal sample. We stop transcription, we filter out the particulate matter, and then we do RNA and DNA preps of all of our two-component system plasmids and their messenger RNA outputs. And then we do next-gen sequencing and ask the question, which two-component systems turn on in the fecal samples, right? And so this is a lot of numbers. Don't worry about the numbers. These are all de-identified at this point. We don't have any gene names here. But on the vertical axis, you're looking at a different human subject. This is a different person from which the fecal sample came. And then the vertical axis is a different two-component system. So there's about 90 two-component systems here we're looking at, and 9 human subjects. And then the color, red versus blue, red means that the two-component system turned on gene expression in that subject's fecal sample relative to growth in normal media. And blue means it turned off, right? And so one thing you see right away is there's about a dozen or so two-component systems here on the left side of the plot that they're red in all 9 or nearly all 9 subjects' fecal samples, meaning they reproducibly turn on gene expression when you treat them with fecal sample compared to growth media. And then you similarly have about a dozen or so on the right-hand side, which reliably turn off in the presence of fecal samples, right? And so we don't know what these are sensing yet. In the fecal sample, they could be sensing general signals like osmolarity, something like that. They could be sensing specific signals, chemical ligands, and so on. We don't know yet, but where we, there's a number of things we want to do with this. One is to screen for two-component systems that sense gut-linked diseases, right? So that a very large number of diseases manifest themselves as changes in the environment of the large intestine of humans, right? And so, or even the small intestine in some cases. And so we're gearing up to collect samples from healthy patients, as well as patients with diseases like ulcerative colitis or Crohn's disease or other diseases. And repeat that screen in order to identify two-component systems that only turn on when you're sick, right? And the idea is to make diagnostic gut bacteria that you could eat that could diagnose and produce a visible readout, for example, of if you have a disease or if you're having a pathological state. And eventually, of course, we want to connect those not just to reporters of disease, but actually to treatments of disease. So we'd like our gut bacteria to both sense and treat diseases at the site of the disease in vivo. We think there's a number of advantages to an approach like that. And so we have a whole arm of the lab that's working on that approach and based on this technology. Okay, so in the last few minutes, I'll tell you another method that we have to improve the performance of these sensors once we use them, right? So if you take any kind of biosensor, like a two-component system, and you measure its response to its input stimulus, what you'll find is that some concentration of stimulus is required to activate the sensor, right? And so we can quantify this as the detection threshold. So this parameter K1/2 right here on the plot on the x-axis, that's the amount of input that is needed to activate the sensor by 50%. It's just a convenient way to characterize its sensitivity for its input. Okay, but if you take biosensors from nature, often that detection threshold is not the detection threshold that you need for your application of interest, right? So if you want to sense a disease biomarker, you may actually need to sense it a couple orders of magnitude lower concentrations than the system that you're using to sense it, which evolved for a totally different purpose. Right? And so we were actually working on a project for sensing gut inflammation, and my student Brian Landry was having this problem. We were trying to sense nitrate, which is a gut inflammatory biomarker, using a nitrate sensor, and it wasn't working. It wasn't turning on. So we needed to make it more sensitive. And so Brian asked, is there a general way I could do this for all two-component systems? Not an approach that's specific just to one system, or is going to require lots and lots of work just to fine-tune my one system and then never be useful for another system. Could I find a general way? And so he had a really brilliant insight, I thought, which is, so the sensor histidine kinases of two-component systems are bifunctional enzymes, right? So when they're active, they phosphorylate the response regulator. But when they are inactive, they actually dephosphorylate. They enzymatically dephosphorylate the response regulator, right? So they're like push-pull type systems. And it's the balance between phosphorylation and dephosphorylation that actually determines how much active response regulators in the cell. And that number, how much active response regulators in the cell, actually determines the response of the pathway, right? So Brian asked the question, if we could, mutate sensor kinases and say reduce the phosphatase activity? Could we actually make them more sensitive, make them activate in response to lower concentrations of their inputs, right? And so he sort of went to a well-known model of two-component system function from Mark Goulian at Penn, and he basically varied the phosphatase activity parameter in that model and then simulated how a generic two-component system would respond to its input. And the model says, yes, this should work over a wide range of phosphatase activities. We should be able to sort of move the transfer function around, move the detection threshold to the left, the less phosphatase activity we have, right? Making it more sensitive. And so he went back to NARX, which is the nitrate sensing kinase. And the literature has established some well-characterized mutations in different subdomains of NARX that actually alter the phosphatase activity, but have no effect on the kinase activity. So one of these is C415R. It's in this dimerization and histidine phosphotransferase domain here with these helices. This is a medium strength mutation that has a medium strength reduction on phosphatase activity of this kinase. And the second is D558V. This is over here in the ATP binding domain. This has a strong negative effect on phosphatase activity of this enzyme. So Brian took our NARXL, one of our NARXL systems. This happens to be the one from Subtilis that's rewired. And he characterized the effect of these mutations on the transfer functions. And so what he saw is that the wild type system here in dark blue had it required 750 micromolar nitrate to turn on. But if he introduced this C415R, this intermediate strength mutation, he could reduce that to 22 micromolar while still preserving the overall function of the sensor. And if he introduced the strong mutation, he decreased the detection threshold even further. Right? Now, he did notice that the leakiness or basal activity of the sensor went up as he introduced this really strong mutation, as you might expect. But this sort of was an initial validation that his idea might be right. And so he went on to show this, I think, very beautifully by actually co-expressing the wild type NARX and this NARX C415R mutant from two different inducible promoters in the same cell. And in this experiment, the total, if you add the amount of wild type sensor kinase plus the amount of mutant sensor kinase, we use the same total expression level in every case. So every blue curve of a different shade here has the same total amount of histidine kinase, but the fraction of wild type kinase relative to mutant kinase is changing. So in dark blue, it's 100% wild type kinase. In light blue, it's 100% mutant kinase. And then in every color in between, we're decreasing the amount of wild type. And what this is doing is continuously decreasing the phosphatase activity of this kinase in the cell. Right? So it's like a biochemistry experiment, but done in vivo. And what you can see is he gets a continuous tuning of the detection threshold. So he's able to sort of match the detection threshold to different levels of nitrate that he might want to sense by just tuning this phosphatase activity. So this, I thought, was a nice validation of his theory that this would work. And then we wanted to be able to do this in general for other two-component systems, right? And so one of the mutations that he used was actually in this so-called GXGXG motif of the NARX sensor kinase. And the first X here, the first variable residue, can be anything in this motif if your two-component system has it. And we looked, and about 65% of sensor kinases have this motif, right? And so we wondered if we couldn't mutate that residue around, and maybe that's affecting phosphatase activity in other two-component systems, right? And so we took a system with no known mutations that no one had ever introduced, because we had just discovered this system a couple of years before. And this is a tetrathionate sensing two-component system. It has a GXGXG motif. And he went in and introduced all 19 mutations at that X site. And he saw that he could vary the detection threshold of the pathway by introducing mutations. In particular, hydrophobic amino acids are tolerated there, and he can tune the sensitivity of the system. And then he repeated this result for another uncharacterized two-component system with no mutations. So we found a hotspot that you can mutate in a two-component system to change its detection threshold for its input, which can be useful if you want to use it as a biosensor for various applications. So he just showed that by using Bacillus subtilis to detect the amount of nitrate in the soil at very low levels. Okay, so what I want to say for this crowd is that we have some nice stories bubbling up now in the lab on using these technologies I just talked about to discover two-component systems, small molecule inhibitors of two-component systems that are involved in virulence and antimicrobial resistance in MRSA, methicillin-resistant Staph aureus, and other microbes. And those stories are about ready to present publicly. So I hope you'll invite me back and I can speak about that, which I think might be exciting to the crowd. We're also using these technologies to characterize how peptides, in particular antimicrobial peptides, interact with bacterial two-component systems that have evolved to sense those peptides and respond by mounting antimicrobial defenses. Right. So in particular, we've developed the first high-throughput method for characterizing peptide two-component system interactions. We've applied this to the classical peptide sensing two-component system FOPQ from Salmonella and other enteric pathogens. And we've made a lot of discoveries of completely new human antimicrobial peptides that are activating this two-component system. And these have implications for actually the role of this two-component system in allowing Salmonella and related pathogens to infect, survive inside of macrophages and mount their antimicrobial responses. Okay, so with that, I'll stop, and I think I thanked most of the people who did the work along the way. And if we have time, I can take questions. Thanks.
Thank you, Jeff. This was fantastic. We only have like 3 minutes for questions, so it's going to be difficult. If anybody has a pressing question, please type it. I was just going to ask you about the interactions, particularly with some of the two-component systems. Sometimes there are accessory proteins, particularly at the membrane. Is there any of these approaches, or are there any of these approaches available to identify how the sensitive and the transduction signal used when an additional component of the system is added to this instead of 2-3 component?
Yeah, that's a great question. And so, yeah, you can often identify by genome sequence that there is an accessory protein, in the same operon as the two-component system. And those accessory proteins have some, conserved features and so on. So yeah, we do have a method to look at that. So, once we port the two-component system into E. coli or subtilis to study it, what we can do is use an inducible promoter to induce a putative accessory protein, right? And so we can induce it over, say, 1,000-fold range. And sometimes what you'll see is if there is an interaction between that accessory protein and the two-component system, it will actually stimulate or deactivate the two-component system, even if the ligand is not present, right? So just the process of overexpression can be good enough to give you a good sense that it might be talking to your kinase and your pathway.
Any other question? Okay, Jeff, this has been wonderful. I'm sure you will receive several emails forward, yep, because I think there's a lot of interest on what you're doing, particularly with my group as well. So we'd love to talk more. Thank you very much. It was wonderful, and I'm sure we will invite you back. Thank you.
Very, very good. Thanks. I was happy to be here. Bye.